So even though this section is certainly focused on a hard science, I like to start by thinking about the complexity of the brain. We're going to begin by focusing on single neurons, and as you know from reading the textbook already, the brain is made up of about 100 billion neurons. Now, that number doesn't really make a whole lot of sense. It's too big to really get our heads around. But when you think about that it's roughly equal to the number of stars in our galaxy, it takes on a manner of perspective that helps us to understand its complexity. Imagine if all of those suns could talk to each other in our galaxy, could communicate, and could emergently produce some consciousness. That's what's happening in our brain every moment. All of those 100 billion neurons are influencing each other, some positively, some negatively, some both, and they're producing patterns of activity that give rise to everything that we are. We're going to start by looking at individual neurons, and then build our way up to looking at the complex network of communications that takes place within the brain. It all begins with a single neuron. Neurons, like other cells, begin with a cell body. The soma, or body of the neuron, contains the nucleus and all of the genetic information of the cell. This is the cell's powerhouse. This is where most of the cell's metabolitic functions take place. Usually on one end of the cell, you have branching extensions called dendrites. These dendritic extensions reach out to receive inputs from other neurons that are nestled nearby. Moving away from the cell body is an axon. You can think of the axon like a nerve bundle, and in fact, a nerve bundle is a set of a whole bunch of different axons moving together through the body. The axon is the chain that links the cell body and the things that the dendrites have been receiving from other neurons to the extension of that neuron down at the end, the terminal branches of the axon. This axon can be very short, or it can be extremely long, as in the case of some motor neurons, like the ones that make up the sciatic nerve. At the end of the axon are the terminal buttons of the axon. These buttons will lie very, very close to the dendrites or soma or other parts of other neurons, and we will use those terminal buttons to communicate with those other neurons. You can always use your hand to think of the parts of a cell. You have the cell body as your palm, your fingers make up the dendrites reaching out to other neurons, and your arm makes up the axon. Neurons function electrochemically. What's so interesting about neurons is that they sit at rest in some tension. There's a gradient between the inside and the outside of the cell, an electrochemical gradient, that keeps the inside of the cell at roughly -70 millivolts relative to the outside of the cell. Now, if you think back to your basic understanding of physics, any kind of gradient will produce a force across that gradient. So there's a charge between the inside and the outside of the cell. That electrical potential is what produces all of the activity inside the brain. Neurons communicate by firing an action potential. What we mean by that is that neurons change their electrochemical makeup over the course of a very short period of time, and that change propagates over the cell and then can be transmitted to other cells. As I mentioned previously, most cells sit at about a -70 millivolt differential at rest. This resting potential produces sort of a tension, like pulling back a spring or a rubber band, that's just waiting to be released. Other neurons communicate with our resting neuron and either excite it by depolarizing the neuron, moving that cell closer to 0 millivolts, right, making it less negative, or by inhibiting the neuron, by making it more negative and moving it further away from 0. Once the cell receives enough excitatory inputs to depolarize it to about roughly -55 millivolts, the cell fires an action potential. What that means is that little gates all across the neuron are triggered to open at a certain change in electrical potential. Once the cell reaches that threshold, what we call the threshold of excitation or the stimulus threshold, those channels open, and suddenly that electrical gradient is allowed to operate. There's a force across that gradient through the ion channels, and negatively charged ions move out of the cell into the extracellular fluid, and positively charged ions move into the cell because there's a force attempting to equalize that electrostatic pressure. What you're seeing in this graph is how quickly that polarization changes. The cell goes from that negative 55 millivolts all the way up to about positive 30 millivolts on average over the course of 1 or so milliseconds. It's incredibly quick, and it's an all or nothing response. Once those ion gates open, there's no going back, and the cell will fire an action potential. Once the cell is maximally depolarized at the peak of its action potential, other factors begin to work to restabilize the cell and return it to its hyperpolarized state. During that refractory period, the cell cannot fire again, so no matter how much excitatory input the cell gets, nothing can happen because you need that electrostatic gradient, that negative resting potential, in order for the cell to work at all. It's sort of a reloading period where nothing can happen no matter how many times you pull the trigger. The cell then resets and returns to resting potential and can receive inputs from adjacent neurons again and can fire again once it reaches that resting potential. So you're seeing that this is a complicated process. Other neurons release chemicals, which we'll talk about in a moment, that either excite or inhibit the receiving neuron. Once that receiving neuron is excited enough to reach that threshold of excitation, it produces an electrical response based on the ions flowing into the neuron, and that electrical current travels down the length of the axon, much like any other electrical wire. And in fact, many axons, particularly the longer ones that work your muscles, are sheathed in something called myelin, a fatty tissue provided by glial cells that help insulate the electrical current. In fact, there's some diseases that cause demyelination, like MS, and you can see the effects of that through jerky movement, difficulty with fine motor skills, and all sorts of other things. Myelin plays an important role in the speed of this electrical process. The faster we are at this electrical process, the faster we are in our fine motor control and our gross motor movements. That electrical potential moves down the axon and finally reaches the terminal buttons of the axon. And this is where that electrical action potential is reconverted into a chemical message to other receiving neurons. Here at the bottom of an axon in a terminal button, we have synaptic vesicles. And the synapse, remember, is just the gap between the terminal button of the axon and the dendrite or the cell body of the next cell that this axon is attempting to communicate. These synaptic vesicles contain lots of molecules of different kinds of neurotransmitters. These neurotransmitters are just chemicals that your neurons use to communicate with one another. When an action potential reaches the end of the terminal button of the axon, this vesicle fuses with the outside of the cell and releases those chemicals, those neurotransmitters, into the synaptic cleft, into this tiny, tiny space between cells. This chemical floats in the extracellular fluid and ultimately attaches itself to receptor sites on the receiving cell. If there are no receptor sites, that neurotransmitter has no effect. And different types of receptors can make the same chemical have a different effect on the receiving cell. So you can see how complicated this process can become. The neurotransmitter that binds to the receptor might have an excitatory or inhibitory effect on the other cell, or it might do all sorts of other things to the receiving cell. But not all of that neurotransmitter is picked up. Some of it hangs out in the synaptic cleft. Usually there's a reuptake mechanism on the cell, sort of like a recycler. So those chemicals can float around in the synaptic cleft, bind to a receptor on its releasing cell, and then get reabsorbed and reused in the next action potential. There are also enzymes like monoamine oxidase that eat up the extra chemicals in the synaptic cleft. And often we use different kinds of pharmacological interventions, different kinds of drugs, to manipulate how much of these neurotransmitters are present in the synaptic cleft. You're probably familiar with SSRIs, or selective serotonin reuptake inhibitors. What these drugs do is block the recycling or reuptake of these chemicals, thereby increasing the amount that's in the synaptic cleft, and more of it floating around means more of it will bind to receptors, and more of it will have an effect on the receiving neurons. This is just a list of a few of the neurotransmitters that are listed in your text, as well as a couple of others that I wanted you to be aware of. These neurotransmitters operate all over the body in the central and peripheral nervous system, and at different places within the nervous system, they have different roles. Acetylcholine, for instance, is responsible for memory. People, for instance, who suffer from Alzheimer's dementia, are given a drug that inhibits the enzyme that eats up acetylcholine, thereby increasing the amount of acetylcholine they have and reducing their symptoms. But it's also responsible for every neuromuscular connection in your body. When a muscle contracts, it's because a neuron is spitting acetylcholine onto the surface of that muscle fiber. Glutamate and GABA are two neurotransmitters that I'd like you to know something about. When I talk about how cells tend to excite or inhibit other cells, what I'm usually talking about are glutamate and GABA. Glutamate is the major excitatory transmitter of the nervous system, and GABA is the major inhibitory neurotransmitter. I won't spend much time discussing the organization of the nervous system in this video, but I do want you to be aware of how the nervous system is organized. The central and peripheral nervous system serves very different roles, but the peripheral nervous system is often overlooked in human psychology. We're going to take a moment and talk about the two components of the autonomic nervous system because I find them so fascinating and because they control so much of our daily lives. The autonomic nervous system is divided into two subsections, the sympathetic and the parasympathetic nervous systems. These two systems essentially control our arousal state. And arousal is an incredibly important concept for any living creature. It helps us control where our resources are being spent. If we spent our lives in a constant state of arousal, our lives would be very short and stressful and difficult. Our calm state is much more valuable for long-term maintenance, but we need to be able to act quickly and act effectively in high-stress situations. So the body is able to turn on and turn off these sort of high-stress, high-activity responses through the sympathetic and parasympathetic nervous system. When the sympathetic nervous system is active, you can think of it as being ready for the 4 F's. You've probably heard of fight or flight, feeding, and the fourth is reproduction. You can tell if the sympathetic nervous system is active in someone just by observing them. Their pupils are dilated. Their breathing is rapid, usually pretty heavy. But if you could get even closer, you'd find that they have a racing heartbeat and that their internal organs have slowed down so that blood can go to the muscles instead of the organs. When the parasympathetic nervous system is active, our body is in a relaxed state. The pupil is constricted, the heart rate is slower. More blood is being sent to the gut for digestion. All of this amazing complexity happens in sync, and it happens automatically without any conscious thought. Most of it's controlled by the brainstem. The fact that our bodies rise to the occasion, even when we don't consciously perceive threats, is really interesting and valuable for understanding how some of our higher level neural processes work when we're under the influence of these systems. For instance, when our sympathetic nervous system is active and we're flush with cortisol, we might make decisions differently than we would if our parasympathetic nervous system was active. I mentioned that neurons fire over the course of just a millisecond. But there's also a much slower and much longer acting chemical messenger system in the body, the endocrine system, or the hormone system. In fact, many neurotransmitters, those chemicals that are released in the synaptic cleft between neurons, are also released into the bloodstream and have a hormonal effect, a longer lasting effect. Epinephrine, or you may have heard called adrenaline, works both between neurons and in the bloodstream. I won't spend a lot of time on the endocrine system, but I do want to point to one interesting example of the endocrine system at work and how long it can take. Have you ever been cut off in traffic or upset by your partner just before you went to work or school for the day? You might have noticed that over the next few hours you remain edgy and frustrated, even if you're not actively thinking about the offense anymore. That might be the result of cortisol. You see, under high stress situations, our body tells our adrenal cortex to release cortisol. Cortisol is released into the bloodstream, and it takes about 3 hours for that stress hormone to go down. When we're under the influence of cortisol, we tend to feel that sympathetic nervous system arousal more easily. So these hormonal systems can have longer-term effects over the course of several hours, even to a single stimulus. But let's spend our time talking about the brain. This is mine, an fMRI image that was taken a couple of years ago. What you're seeing here is called a mid-sagittal slice. The brain cut directly in half and looking straight in at the side. You can actually see several structures. The first and the most prominent is the corpus callosum, that curved structure right in the middle of the brain. This is the bundle of fibers linking the left and the right hemisphere of the brain. You can also see a cauliflower-shaped structure down in the bottom left. That's the cerebellum, part of the hindbrain, responsible for balance and other sort of fine motor movement. The brainstem extends just down below the corpus callosum and between the cerebellum and the corpus callosum, and you can see it intersecting with the spinal column. All of that sympathetic and parasympathetic activity we were talking about takes place in this area. We're going to get into the anatomy, but just before we get into the anatomy, I want to draw your attention to how we can see this. There are several different ways that we image the brain, and we're getting better and better at it all the time. In fact, the most powerful magnets in the world were just unveiled in the United States, and we should see some really interesting images of living brains using those magnets. You should take a careful look in your book at the different methods of imaging. I just want to talk for a moment about fMRI. What functional magnetic resonance imaging does is allows us to see the blood response in the brain over a very specific period of time. For instance, if I show you the picture of a red ball, and I measure your blood response in the brain, I can see very carefully which parts of the brain are requesting more blood. If they're using more blood, they're using more glucose, and if they're using glucose, they're firing. So we know that those parts of the brain are what's active when we're looking at a red square. This becomes really interesting if I can differentiate how the brain responds to a red square versus a red sphere, or a blue square. If I can identify different parts of the brain that respond to different things, I might be getting at the functional part of that particular area of the brain. In this image, you can see some of the things labeled that you saw in the earlier image. Just below the corpus callosum is the thalamus. This part of the brain is our sensory hub, where all of the primary sensory cortex, where your eye projections go, where your auditory projections go, where your olfactory bulbs go, they go straight to the thalamus. And your thalamus is the first area that processes that sensory information. The thalamus is an older part of the brain. Most animals have some kind of sensory hub like our thalamus. And in fact, many sensory decisions are made there before we're even consciously aware of it. Wrapped around the thalamus is the hippocampus, called so because it's sort of shaped like a seahorse. The hippocampus is critically responsible for our learning and memory. I mentioned a patient, HM, who could no longer form new memories. The reason he couldn't form new memories is because of hippocampal damage. The hippocampus is responsible in forming memories, but it doesn't seem to be where memory is stored. This older part of our brain also seems to contain most of our emotional processing. The cingulate cortex, the first layer of cortex, which wraps around the corpus callosum and is highly integrated with the thalamus, is responsible for most of our emotional processing. Sitting atop all of that older brain structure is the neocortex, or new cortex. We call it new because it's new from an evolutionary perspective. As you can see here, the neocortex is divided into 4 lobes: the frontal lobe, the temporal lobe, the parietal lobe, and the occipital lobe. Each of these lobes perform different functions and are geographically separated by several gyri and sulci. You can think of the gyri as the mountains and the sulci as the valleys between the folds in the brain. We'll start by talking about the parietal lobe. The parietal lobe includes secondary sensory cortex. So after material gets processed by the sense organs and sent to the thalamus, the parietal cortex is where all of that sensory information gets put together and allows us to connect sights and sounds together. Behind the parietal cortex, at the very back of your brain, is the occipital lobe. The occipital lobe is almost entirely used for processing vision. Vision is our largest and most important sense, and it takes a lot of cortical area to perceive the depth of color that we see, for instance, and to orient things in 3-dimensional space. On the sides of your brain is the temporal cortex. The temporal cortex is responsible for a variety of things, but most notably memory, language, and the sense of hearing. For example, there are specific areas of the temporal lobe that we know that are responsible for recognizing certain types of objects. The fusiform face area of the fusiform gyrus is responsible for recognizing faces. People with damage to the fusiform face area are unable to recognize the faces of individuals that they know. The frontal lobe is the newest part of the cortex, and the frontal lobe is responsible for executive planning and inhibition. Inhibition is an incredibly important part of the brain's role. Inhibition is what prevents us from going after what we immediately want in the immediate moment. It's what prevents you from picking up that banana at the grocery store and just eating it there in the aisle. This frontal inhibition is incredibly important for social living, and as such, the frontal lobe and damage to the frontal lobe are particularly interesting areas to study in psychology. Right between the frontal and the parietal lobe lie two very important strips of cortex, the primary motor cortex and the primary sensory cortex. These strips control all of the movement and the senses from our body. What's interesting about the motor and sensory cortex is that they're topographically organized. That is, we can map the certain parts of the motor cortex that are responsible for different parts of the body. In fact, during open brain surgery, you can manipulate different parts of this cortex electrically and cause movement in an individual. What you can see in the bottom left of this image is what we call a motor homunculus. This is what the human body would look like if the ratio of brain tissue to body tissue were 1:1. That is, if the areas of our body that get the most brain tissue also got the most physical space. As you can see, the hands are huge. The hands take up the largest portion of our motor cortex. Also very large are the lips, face, and tongue. You can see that the parts of our body where we have very little motor control, for instance the back of our arms or the back of our shoulders, are very small. The tops of our legs and the ankles, very small, because they don't use a lot of motor control. They don't have a lot of muscle. A sensory homunculus would look similar, but not the same. You'll notice a lot more space for the face itself and slightly less emphasis on the hands. You can see in the image on the right how this cortex is organized. One of the other terribly interesting things about the brain is its contralateral organization. That is, the right half of the brain controls the left half of the body. We know this through a variety of studies, but most interesting are the split brain studies. If the corpus callosum is severed, there's only a few other places where the two hemispheres talk to one another. That is, most of the communication on one half of your brain is isolated on that half of the brain without the corpus callosum. Most things are shared across both sides of the brain. However, language in particular tends to be isolated to the left side of your brain. So if your left and your right side of the brain are severed, your right half of the brain has a difficult time accessing your speech. That is, it's very hard for you to articulate something that only the right half of your brain has seen. How do we know that? Well, take a look at this video. I realize that's a little old, but I find that video fascinating because it goes to show how much of what goes on in our brain isn't necessarily accessible to language. And if it's not accessible to language, it's not accessible to our consciousness. So how much of our lives take place outside of our conscious awareness? It's worth mentioning here when we talk about the right and left half of the brain, that all of the things that talk about whether you're left-brained or right-brained are total bunk. They're pseudoscience, non-scientific, as is any idea that we use less than 100% of our brains. We may not know what everything's function is, and some people may be more linguistically dominant than artistically dominant, but that doesn't mean anything is functioning differently in our brains. The brain is a fascinating piece of architecture. It's a galaxy inside our heads. It has perhaps the most power of anything we know that exists in the world, but it may not have enough power to fully understand itself. Every day, neuroscientists are making new leaps and bounds in knowledge and learning new things about the brain. And over the course of your lifetime, even over the course of this term, new things will be discovered about the way the brain functions. I'm curious as to your thoughts on the brain. Can we be reduced down to the simple firings of neurons? Is that all we are? Or is there something more than the emergent processes of these biochemical functions?